Bismillahirrahmanirrahim. In this video, I am going to discuss the morphology and functional characteristics of smooth muscles. Let's begin. Smooth muscles have been given the name because the cells do not have striations. Smooth muscles are present in the walls of hollow organs like the urinary bladder, uterus, stomach, intestine, and in the walls of passageways such as the arteries and veins of the circulatory system, and in the tracts of respiratory, urinary, and reproductive systems. Smooth muscle is also present in the eyes, where it functions to change the size of the iris and alter the shape of the lens, and in the skin, where it causes hair to stand erect in response to cold temperature or fear. Smooth muscle fibers are spindle shaped and have a single nucleus. They are thousands of times shorter than skeletal muscle fibers. Smooth muscles produce their own connective tissue, the endomysium. Smooth muscle fibers do have actin and myosin contractile proteins and thick and thin filaments, but these are not arranged regularly as sarcomeres. The thin filaments are anchored by dense bodies. A dense body is functionally similar to the Z discs of skeletal muscle fibers and is fastened to the sarcolemma. Smooth muscles also contain intermediate filaments. Smooth muscle is not under voluntary control, thus it is called involuntary muscle. The triggers for smooth muscle contraction include hormones, neural stimulation by the autonomic nervous system, and local factors. In certain locations, such as the walls of visceral organs, stretching the muscle can trigger its contraction. Axons of autonomic nerves do not form the highly organized neuromuscular junctions with smooth muscle, as seen between motor neurons and skeletal muscle fibers. Instead, there is a series of neurotransmitter filled bulges called varicosities. These loosely form the motor units. A varicosity releases neurotransmitters into the synaptic cleft. Because smooth muscle fibers have a much smaller diameter than skeletal muscle cells, T-tubules are not required to reach the interior of the cell, and therefore not necessary to transmit an action potential deep into the fiber. The sarcoplasmic reticulum in smooth muscles are poorly developed. Due to this, smooth muscle fibers have limited calcium stores. To compensate, smooth muscles have calcium channels in the cavuli that open during the action potential along the sarcolemma. Cavuli are membrane indentations in the sarcolemma. The influx of extracellular calcium ions accounts for most of the calcium that triggers contraction of a smooth muscle cell. In a smooth muscle fiber, external calcium ions passing through opened calcium channels in the sarcolemma and additional calcium released from sarcoplasm cause a rise in the calcium levels in the sarcoplasm. It is important to note here that smooth muscle cells do not have troponin. Hence, cross-bridge formation is regulated by a regulatory protein called the calmodulin. Increased sarcoplasmic calcium binds to calmodulin and activates it. The activated calcium calmodulin complex then binds to an enzyme called myosin light chain kinase and activates it. Myosin light chain kinase activates the myosin heads by phosphorylating them. During this process, myosin kinase converts ATP to ADP and inorganic phosphate, with the inorganic phosphate attaching to the myosin head. The myosin heads can then attach to actin binding sites and pull on the thin filament. When the thin filaments slide past the thick filaments, they pull on the dense bodies. Structures tether to the sarcolemma, which then pull on the intermediate filaments network throughout the sarcoplasm. This arrangement causes the entire muscle fiber to contract in a manner whereby the ends are pulled towards the center, causing the midsection to bulge in a corkscrew motion. Muscle contraction continues until ATP-dependent calcium pumps actively transport calcium ions back into the sarcoplasm and out of the cell. This causes inactivation of calmodulin and inactivation of myosin light chain kinase. To relax a contracted muscle, myosin must be dephosphorylated. This dephosphorylation is mediated by the enzyme myosin light chain phosphatase, which is continuously active in smooth muscle during periods of rest and contraction. When cytosolic calcium concentration increases, the rate of myosin phosphorylation by the activated kinase exceeds the rate of dephosphorylation by the phosphatase, and the amount of phosphorylated myosin in the cell increases, producing an increase in tension. When the cytosolic calcium concentration decreases, the rate of phosphorylation decreases below that of dephosphorylation, and the amount of phosphorylated myosin decreases, producing the relaxation. Because most smooth muscles must function for long periods without rest, some smooth muscle can maintain contractions even as calcium is removed and myosin kinase is inactivated. This can happen as cross bridges between myosin heads and actin keep the thick and thin filaments linked together for a prolonged period and without the need for ATP. This phenomena is called the Latch-bridge mechanism. It maintains isometric tension in an almost rigor-like state without movement. The Latch-bridge mechanism allows for maintaining of muscle tone in smooth muscle that lines arterioles and other visceral organs with very little energy expenditure. Another good example of the usefulness of this mechanism is seen in sphincter muscles of the gastrointestinal and urinary tracts, where smooth muscles must maintain contraction for prolonged periods. The ATPase activity of smooth muscle myosin is 10 to 100 times less than that of skeletal muscle myosin. Because the rate of ATP hydrolysis determines the rate of cross-bridge cycling and shortening velocity, smooth muscle shortening is much slower than that of skeletal muscle. Due to this slow rate of energy usage and added advantage of latch bridge mechanism, smooth muscle are highly energy efficient and do not undergo fatigue during prolonged periods of activity. The smooth muscles can be placed into one of the two groups based on the electrical characteristics of their plasma membrane: single unit smooth muscles and multi-unit smooth muscles. The two types have different locations in the body and have different characteristics. Single unit muscle has its muscle fibers joined by gap junctions so that the muscle contracts as a single unit. This type of smooth muscle is found in the walls of all visceral organs except the heart, and so it is commonly called visceral muscle. The visceral smooth muscle exhibit a property called the stress relaxation response. This means that when a hollow organ is filled, the smooth muscle of the organ is stretched. The mechanical stress of the stretching triggers a brief contraction, but this is immediately followed by relaxation so that the organ does not empty its contents prematurely. This is important for organs such as the stomach or urinary bladder. When the hollow organ is empty, the contraction of smooth muscle causes shrinkage to avoid flabbiness. Multi-unit smooth muscles do not possess gap junctions and thus are not electrically coupled. As a result, contraction does not spread from one cell to the next, but is instead confined to the cell that was originally stimulated. Stimuli for multi-unit smooth muscles come from the autonomic nerves or hormones, but not from stretching. This type of tissue is found around large blood vessels, in the respiratory airways, and in the eyes. Some types of smooth muscle cells generate action potentials spontaneously and in the absence of any neural or hormonal input. The plasma membranes of such cells do not maintain a continuous resting membrane potential. Instead, they gradually depolarize until they reach the threshold potential and produce an action potential. Following repolarization, the membrane again begins to depolarize, so that a sequence of action potential occurs, producing a rhythmic state of contractile activity. The membrane potential change occurring during the spontaneous depolarization to threshold is known as a pacemaker potential. Pacemaker cells in other smooth muscles have a slightly different pattern of activity. The membrane potential drifts up and down due to regular variation in ion flux across the membrane. These periodic fluctuations are called slow waves. When an excitatory input is superimposed, slow waves are depolarized above threshold and action potentials lead to smooth muscle contraction. Pacemaker cells are found throughout the gastrointestinal tract. Thus, gastrointestinal smooth muscle tends to rhythmically contract even in the absence of neuronal stimulation. Some cardiac muscle cells and some neurons in the central nervous system also have pacemaker potentials and can spontaneously generate action potentials in the absence of external stimuli. Thanks for watching and listening. Do share this video with your friends and colleagues. If you like this video, subscribe to this channel for watching more videos.